Welcome to today's lesson on the use of stem cells in medical treatments, where we will explore the potential benefits and challenges associated with using these remarkable cells in medicine. In our lesson today, we will discuss how stem cells are utilized in medical treatments, including therapeutic cloning. Additionally, we will cover the potential issues surrounding the use of embryonic stem cells and study the broader ethical and practical concerns related to the use of stem cells and embryos in medicine. So far, you have learned that a stem cell is an undifferentiated cell, capable of developing into different cell types. You are also familiar with the concept of embryonic stem cells and adult stem cells. This foundational knowledge allows us to appreciate the versatility and importance of stem cells in medical research and treatments. Understanding the use of stem cells in medicine advances our biological understanding by highlighting their potential to treat diseases, repair damaged tissues, and offer new avenues for regenerative medicine. By exploring these concepts, we gain insights into future possibilities for medical innovation and the ethical considerations that accompany such advancements. To begin, let's remind ourselves what stem cells are. Stem cells are unique because they have the potential to develop into many different cell types in the body during early life and growth. Unlike specialized cells such as nerve or blood cells, stem cells can replicate themselves numerous times or generate new cells through differentiation. Stem cells have significant potential in treating various diseases and conditions due to their ability to generate new cells and tissues. In people suffering from diabetes, stem cells can be used to replace damaged or non-functional insulin-producing cells in the pancreas. This could potentially reduce or eliminate the need for insulin injections. For individuals who are paralyzed due to spinal cord injuries, stem cells can be used to generate new nerve cells, helping to repair damaged spinal tissues and potentially restore movement and sensation. Stem cells can also be used in therapeutic cloning. Therapeutic cloning is a technique that involves creating an embryo with the same genetic material as the patient. Here's how it works. First, the nucleus, containing genetic material, is removed from one of the patient's cells. The nucleus is then also removed from a donor egg, and the empty cell is kept. The patient's nucleus is then inserted into the egg cell from the donor. Then the egg cell is stimulated to develop into an embryo. Stem cells are then harvested from this embryo and can be grown into the required types of cells, tissues, or organs. The primary advantage of therapeutic cloning is that the stem cells generated share the same genes as the patient, which minimizes the risk of the patient's body rejecting the new cells, tissues, or organs. We have discussed the serious benefits of stem cell therapy, including for treating diabetes and nerve damage, and the reduced risk of infection. Despite their promise, the use of stem cells in medical treatments comes with several potential issues. First is the risk of contamination. Stem cells could become contaminated with viruses or other pathogens. If these contaminated cells are used in treatments, there is a risk of transferring infections to the patient. It's also important to note that some people have ethical or religious objections to the use of human embryos in stem cell research. They may believe that creating and using embryos for research and treatment is morally unacceptable. Furthermore, stem cell research and treatments can be challenging and expensive. Some people argue that the financial and time resources invested in stem cell research could be better spent on other medical research and healthcare services. Because of this, understanding the potential of stem cells in medical treatments and their associated challenges is crucial. On one hand, stem cells offer the possibility to treat and even cure certain debilitating conditions. On the other hand, we must navigate the ethical, financial, and biological hurdles that come with their use. In today's lesson, we focused on the role of stem cells in medical treatments, including therapeutic cloning. We began by defining stem cells and discussing their unique ability to differentiate into various cell types, making them invaluable in regenerative medicine. We explored the medical uses of stem cells, such as replacing damaged or diseased tissues with healthy ones generated from stem cells. Therapeutic cloning was also discussed, where an embryo is cloned to harvest stem cells that are genetically identical to the patient, minimising the risk of immune rejection. Furthermore, we examine the practical issues associated with the use of embryonic stem cells, as well as possible ethical issues that arise. We also learnt about balancing the advantages and disadvantages of stem cell treatment. By understanding these aspects, you can gain a comprehensive view of the promises and perils of stem cell technology and its profound impact on modern medicine.
Watch the rest of the GCSE biology course at Sophos Education.